Our final video of the biochemistry unit is going to be all about enzymes, those special proteins that we briefly mentioned in our last video. So enzymes are proteins. That's very important to remember. They're a special type of protein. So their shape is very important. Remember, if they denature, they won't work anymore. And the shape is very, very critical to enzymes. But these are specialized proteins with very specific shapes that speed up chemical reactions. They're very important because without enzymes, pretty much every single reaction in our body wouldn't happen. We have tons of reactions going on in our body at all times, and almost all of them are being mediated or helped out by these enzymes. There's many of the reactions that we do where if we left them on their own, they would almost seem to never happen because they were so slow. So these are highly important to being a functioning human being. And you often hear them used when we talk about digestion. Oh, our enzymes are helping us digest our food, but they're actually working all the time in our body in every single part. Their names often end in ASE, things like lactase that helps us break down the lactose in milk. So if you don't have this one, you'll probably be lactose intolerant. Primase and helicase, these are all things that are going to help us copy our DNA, and we'll learn all about that down the road. So the key point that I want you to remember for this whole thing is that enzymes are never used up in a reaction. They never completely disappear. They never break apart when the reaction is done. They're reused again and again. I like to think of it as like a cookie cutter. Just because I cut out one cookie doesn't mean my cookie cutter breaks. I can then pick it up and go cut out another cookie, and then another, and then another, and I still keep the same shape, and I can still do the same job over and over again. Now, like I said, the shape of an enzyme is very, very important. So their shape helps them do their job. The enzyme itself has what we call an active site, this part here that I'm drawing on. This active site is the place on the enzyme where the substrate will bind and the reaction will occur. Whatever reaction we're trying to speed up, it's going to happen right there in the active site. So what is this substrate that I mentioned? Well, the substrate is this purple guy here. It's whatever molecule or two molecules that we want to do the reaction on. So this is the chemical, the compound that we want to either break apart or the two compounds that we want to bring together in our reaction. So it's going to fit inside the enzyme's active site perfectly, and it's going to have the reaction happen to it. It's just like a lock and a key. The substrate is going to have a very specific shape as well, and its shape fits the enzyme's active site perfectly. So you can think of the key as the substrate and the lock as the enzyme. It's going to fit in there, and only one key can work in that lock. I can't take a different substrate and try to put it in that enzyme because it just won't work. They don't fit together. So why this is important is because I can have many different reactions happening at the same time with many different enzymes, and I don't have to worry about one of them bothering or messing up the reactions of another. So how does this all work? We've learned the overall structure of it, but what's going on? So first let's look at this picture here. We have our little enzyme in purple, and we have our little substrate here in green. They're going to come together, and our substrate is going to fit inside the active site of our enzyme. The reaction is going to happen, and in this case, the enzyme is going to break that bond connecting the two pieces of our substrate, and then those two pieces are going to then leave the enzyme, and our enzyme is going to start all over again. So this time we went from one substrate and we broke it to make two substrates, our products. So the substrate will bind to the active site, and then the chemical reaction occurs. Either two pieces will be brought together or one piece will be broken into two pieces. And remember that a chemical reaction happens when different elements and molecules combine to form new substances. So we're making entirely new substances here. We started with something called sucrose, or what you probably know of as table sugar, and we ended with glucose and fructose, the two parts that make up our table sugar. So the substrate is broken down into two new pieces or combined to make a new one. So things like digestion will break down particles, and we'll be taking one big one and turning it into smaller ones. If we want to grow, on the other hand, though, or if we're making new molecules, I want to take smaller pieces and combine them together to make bigger ones. Whatever we make at the end of the reaction is called the product. Remember way back from our first video, we talked about that a little bit, where we said the reactants go in and the products are produced at the end of the reaction. Same thing here. Whatever we make is going to be called the product. So the way that enzymes actually speed up this reaction, though, is because they do something called lowering the activation energy for the chemical reaction. And the activation energy is just the amount of energy that the body or the cell needs in order to get that reaction started. Nothing is free in the world. Everything requires energy to do. So sometimes there's a lot of energy required to get that reaction started. And so it's almost impossible for us to supply enough energy to our bodies to get that reaction going. So the enzyme helps us out and makes us need less energy to get that started. Think about this hill is the amount of energy that we're going to have to put into the reaction. And we want to roll this log up the hill. If it's a really, really big hill, you're going to need to put a lot of energy into that, and you may not even be able to make it if the hill is so big. What an enzyme will do is it will take that normal activation energy and squish it down so that it's a lot less. So think of having to roll the rock, all the way over that hill versus just over this hill. You'd much prefer the smaller hill. You'll be able to get the reaction started faster because there's less energy and less work you have to put into it. So we say that the enzyme is a catalyst, or a chemical that speeds up reactions because it lowers this activation energy. The very last point here, guys, and I already mentioned this one, so this will tell you how important it is. Enzymes are recycled and reused. Just like in that picture with the enzyme when we were learning about the products and how it breaks apart, this is kind of similar. I have my enzyme, it does its reaction, it forms the products, but then it comes back and starts all over again. And it looks exactly the same in all three of these pictures. An enzyme is never changed, it's never destroyed by its chemical reaction that it does. It's recycled and reused. It can be used over and over again for thousands and thousands of reactions. So like I said, think of it as our cookie cutter. The cookie cutter doesn't get destroyed just because I cut one little cookie out. Or think of Pac-Man if you like to play Pac-Man. He can go around eating all of the little white food pellets, and he doesn't change his shape. He's still just going and going through the game. All right, see you guys in class.